Welcome to the webinar on the CometAssay, a technique for quantitating DNA damage and repair from in vivo and in vitro samples of eukaryotic cells. In today's presentation, we'll provide an overview of the methodology for the detection of single and double-strand DNA breaks in single cells. The topics for today's webinar will begin with an overview of the methodology. We'll then switch to an examination of the tools and reagents for performance of the CometAssay in order to achieve reproducible results. These tools and reagents will focus on the Comet control cells and the Comet electrophoresis system. I will then switch to a comparison and contrast of the alkaline versus the neutral CometAssay when examining single versus double-strand DNA damaging agents. So we'll first begin with the overview. The CometAssay is a single-cell gel electrophoresis assay for evaluating DNA damage in cells. The premise of the CometAssay is that damaged DNA is fragmented. And when you increase the DNA damage to the organism, what will result is an increased number of DNA fragments. One then can visualize the different fragment sizes if the DNA is exposed to an electric field. In an electric field, the smaller fragments will move further and faster. And this is applicable for both single and double-strand DNA. There are many applications for use when using the CometAssay. One can study cellular response to DNA damage. You can screen for inhibitors to DNA repair, test chemicals for toxicity, screen for environmental mutagens, and also probe for specific types of DNA damage using DNA repair glycosylases. I'll explain this last point in more detail within the presentation. The Alkaline CometAssay is the most popular protocol to perform and the easiest to run. The Alkaline CometAssay is presented in the top panel. To begin with, a single cell suspension is made, and the single cells are then mixed with low melting agarose at 37 degrees. This agarose cell mixture is then spread onto a pre-treated slide. These pre-treated slides promote the adherence of the LM agarose during alkaline electrophoresis, and these pre-treated slides are critical. Once the LM agarose and cell solution has set, the cells are then treated with a lysis solution. The lysis solution consists of detergent and a high salt solution. The lysis solution will remove the membranes and the histones from the DNA. Lysis treatment can occur for either 1 hour at 4 degrees or overnight at 4 degrees. After the lysis step, the slides are then immersed in an alkaline solution which has a pH greater than 13. At this step, the DNA is unwound and the strands are denatured. It's in this single-stranded stage for which when the DNA is then treated to alkaline electrophoresis. So in step 5, the larger fragments will stay closer to the left edge, which creates the head, and the smaller fragments, which indicate the level of damage, you see migrating to the right. So the resultant form is this teardrop type of conformation of a damaged cell. After alkaline electrophoresis, the slides are neutralized, and then the LM agarose is completely dried down. After drying down, the slides are then stained with a fluorescent dye, allowing for fluorescent microscopy in order to score for the level of damage. Another type of Comet assay to perform is the neutral Comet assay. It is less sensitive, primarily because it's only going to detect double-strand breaks. In the neutral Comet assay, after the cells have been treated with lysis solution, they are then neutralized, and electrophoresis occurs using a neutral buffer, so the DNA strands remain in their double-strand conformation. So the image at the bottom is a resulting comet after neutral electrophoresis, and you can note that the shape is different than what is seen when it's performed under alkaline electrophoresis. You have more of an elongated shape to the comet tail. So to summarize, there's two CometAssay formats. There's the alkaline comet, which is performed under denaturing conditions for the DNA, which is used to analyze all DNA breaks. Second, you can perform the neutral comet. This is performed under non-denaturing conditions, and the DNA remains in its duplex state. So for neutral comet, you're analyzing double-strand breaks. For both assay formats, you can quantify the level of damage. So the damage parameters, which are commonly used in Comet, are percent DNA in the tail and tail moment. Both of these damage parameters focus on the damaged DNA which is contained in the tail. On the figure in the right, when you're calculating the Comet damage parameters, the head region and the tail region will be marked. So to calculate percent DNA in the tail, one would take the integrated tail intensity times 100 and divide by the total integrated tail intensity. So it would be the tail over the whole intensity from head to tail. Tail moment, on the other hand, will measure the severity of the damage. What it combines is the percent DNA in the tail with the distance of migration. When staining the comets, there are choices. One can use a fluorescent dye, which of course will require a fluorescent microscope. In the table listed, there are a variety of different dyes which have been used in the Comet assay. The most popular dye currently is cyber gold. The reason cyber gold is preferred is because if you look at the column where it shows signal to background, you see greater than 1,000 signal to background, which gives wonderful sensitivity. Also, a criteria to consider is that you need to select a dye that binds both the single-strand and double-strand DNA. So in these two columns, you're looking to achieve a ratio which is close to 1. But the primary driving force is you need to see the signal above the background, so cyber gold is the preferred choice. Some customers prefer to be able to archive their samples long-term. So for long-term archiving, one can use silver stain. You can silver stain your Comet slides. In order to first quantify, though, you can always first stain with the fluorescent dye and then come in later with the silver stain. In this slide, these are examples showing different levels, increasing levels of damage, So you can see from samples 1 through 10 in the figures, you can see an increasing intensity in the tail region. So in the table on the left, these images are also shown with the damage parameters, percent DNA in the tail, tail moment, and tail length. So you can see that when damage is increasing in the tail region, then you should also see a subsequent increase in each of these damage parameters. Now, of course, it's always possible to not actually determine precisely the damage parameters. Some customers will simply throw their Comet images into, say, you have groups 0 to 4. 0 would be healthy, 4 would be the highest level of damage. So it is possible to do a visual scoring, but most customers prefer to calculate the damage parameter. This is an example where you have created damage to the DNA, and now you're monitoring recovery through DNA repair. And this is using the alkaline electrophoresis. Jurkat cells were treated with hydrogen peroxide for 1 hour. Then the damaging agent is removed, and then the cells are allowed to recover for 1 or 2 hours. And then samples from each of the populations were then subjected to the Alkaline Comet assay. So if you look at the images on the left, you can see the healthy cells are extremely, have minimal tail, percent damage in the tail. The treated cells show a high level of damage, but then after 1 to 2 hours of treatment, the cells have begun to recover, and you're losing the amount of damaged DNA in the tail. And this is reflected in the damage parameters that's shown in the table on the right. If you look at the cell sample, and we can focus on percent DNA in the tail, the healthy population was showing about 3% DNA in the tail. Immediately after treatment, it was 90% DNA in the tail, indicating a high level of damage. But after 2 hours, the damage parameter has fallen back to 9.4%. And this is reflected in all the damage parameters, showing a reduction with recovery. So there are choices that one can choose for data analysis. The less expensive choice when trying to determine the damage parameter is one can acquire software only. So this is a selection of possible systems that are available with their associated websites. The software only, the customer will have to provide their own microscope, camera, and the computer hardware for acquiring the images. There are also more expensive commercial packages available that include the software for the Comet analysis. These commercial packages are aimed for higher throughput labs to reduce the time for data analysis. We'll now switch to tools and reagents for performing the Comet. Okay, Trevigen offers a standardized CometAssay system. Trevigen offers the instrumentation, the electrophoresis system, and the control cells, which can be used for laboratories to standardize their electrophoresis procedures for reproducible results. We also offer different slide formats and kits and reagents for those labs who are dealing with large number of samples. Trevigen offers 4 types of slide formats. You can a 2-well slide, 3, 20, or 96-well slides. All the slide sets are treated to promote the adherence of the LM agarose and alkaline solution. And this is key when performing the alkaline assay. If the slides are not pre-treated, the agarose disc will detach when the pH is greater than 13. So it's important to always use specially treated slides. The hydrophobic barrier also allows easy application of the cells directly to the slide. And it also allows the microliter addition of the fluorescent dyes directly to the wells for staining. So this decreases the cost of fluorescent staining. The different formats are designed to shorten the assay time based upon the number of samples that a lab is working with. So once a customer is familiar with the CometAssay, there is no difference between running a 2-well slide and a 20-well slide. The electrophoresis conditions are identical. The value of starting to use a 20 and 96-well slide is that you can now reduce the amount of reagent that's being used to process the number of samples. Trevigen offers 3 basic Comet kits. The CometAssay kits come with a detailed manual which provides protocols for the alkaline and neutral Comet assay with fluorescent staining. Within the kit comes the lysis solution, LM agarose, and dependent upon your need, a Comet slide, which can either be 2, 20, or 96 well. The customer will have to purchase the fluorescent dyes separately, but we strongly recommend Cyber Gold. CometAssay Silver is the CometAssay kit, which also includes the silver staining components for permanent archiving of the samples. But as I mentioned before, one can still perform the CometAssay, stain with the fluorescent dye, which is seen in the figure on the top right, which allows quantification of the amount of DNA in the tail, and then come in on the second step and silver stain the exact same Comet images. The third kit offered is the Flare Assay kits. This is a modified alkaline protocol which now is going to include a step where you treat the DNA with a lesion-specific repair enzyme. And in the following slides, I'm going to describe the Flare assay. Not all DNA damage results in DNA breaks. And DNA breaks is a strict requirement for the Comet analysis. So in order to identify specific lesions, there are DNA repair enzymes that are specific. For example, the DNA glycosylases for oxidative damage are FPG, HOGG1, and Endo3. But they all have their different specificity. FPG and HOGG1 recognizes oxidative damage of purines, while Endo3 recognizes oxidative damage of pyrimidines. So the FLARE stands for Fragment Length Analysis Using Restriction Enzymes. So it's a modified alkaline Comet assay where you're increasing the sensitivity of the assay by using enzymes to recognize the damage and create the single-strand nick. In this slide, this is an application of studying oxidative damage. In this instance, Jurkat cells were treated with potassium bromate to create oxidative damage. So if you look at the cell sample and you compare the healthy to the treated sample, in the absence of enzyme, and we're looking at percent DNA in the tail, you can see no difference in the healthy versus the treated sample, 4.3 versus 5. However, if after the lysis step, we pre-treat with DNA glycosylase, which could recognize the damage and create the nick, then you proceed on with normal alkaline Comet, you could increase sensitivity. If you look at the healthy population and you treat with FPG, HOGG1, and Endo3, you can see a dramatic increase in the percent DNA in the tail when the healthy population is treated with FPG. It's gone from 4.3 to 21.3. Now, if we now compare the healthy to the treated population, you can now see that FPG, it was at 21% in the healthy, has also increased in the treated to 33.4%. So it's recognizing additional damage caused by the potassium bromate. HOGG1, however, appears to be more specific. In the healthy population, the percent DNA in the tail is very close to what the no enzyme control was. However, in the treated population, you're seeing an increase from 7 to 13% DNA in the tail. So the HOGG1 is more specific for oxidative damage with the potassium bromate. The Endo3, on the other hand, showed no difference between the healthy and the treated population. We'll now switch to the Comet control cells. The purpose of developing the control cells was to standardize and compare electrophoresis methods between users and laboratories. The alkaline electrophoresis is especially problematic when performing the CometAssay. So we wanted to develop standards to help develop a Comet electrophoresis system. The control cells contain 4 unsynchronized suspension cell preparations with incremental increases in DNA damage. We've developed control cells for the alkaline CometAssay and also control cells for the neutral CometAssay. Both sets of control cells are designed for long-term storage in liquid nitrogen. This is a set of data when using the alkaline control cells in alkaline electrophoresis. So the alkaline control cells are sold in 4 vials. We start with a healthy population of Jurkat cells, which would be CC0, that have been differentially treated with etoposide to increase the level of damage in the population. So CC1, 2, and 3 have all been treated differently with etoposide to increase the level of damage. So the images on the right are some images of the cells within the population where you can easily see an increase in the percent DNA in the tail. The table on the left is a typical box and whisker plot that scientists use to illustrate the data and the spread of the damaged cells within their population. And with alkaline Comet, the most common damage parameter to use is percent DNA in the tail. Now the plot, the mean is indicated by the diamond, blue diamonds, and the medians are the dashed lines within the box plot. And you can see that they're closely aligned, which indicates a well spread of the damage within the population. This is an example of using the neutral control cells in neutral electrophoresis. Again, it's sold as 4 vials in which you begin with a healthy population, which is the NC0, We then treated with a double-strand DNA damaging agent, bleomycin, which induces double-strand cuts. And you see increasing amounts of damage in NC1, 2, and 3. The images on the right, again, in the neutral electrophoresis, you're going to see more the elongated tail images. And again, you can see the increasing amount of damaged DNA in the tail. In the box plot on the left, the common damage parameter which is used with neutral electrophoresis is tail moment. The main reason for this is in neutral electrophoresis, the tail length keeps increasing, but this is not true for the alkaline. So you really need to be using tail moment as the damage parameter for neutral electrophoresis. The control cells are especially valuable when you're trying to set up a new user in the laboratory because you have defined level of damage within the population which you hope to achieve with reproducible results. So as I mentioned, Comet alkaline electrophoresis can be problematic. There is a number of variables that have to be optimized, and this is especially true if you want to compare results between laboratories which are using different electrophoresis units. The parameters that have to be controlled are the pH, sodium hydroxide concentration, the position of the slide in the electric field, the distance between the electrodes, the buffer above the slide during electrophoresis, as well as the time of electrophoresis. So one can see that if you are using two different electrophoresis systems and studying a project, more than one of these variables are going to have to be optimized to compare results. An example of the problem that's seen is that the time of electrophoresis will have to be increased if you start to increase the buffer height above the slide as well as the sodium hydroxide concentration. In the following slides, I'm going to show some of the problems that one can see when you're trying to optimize your own electrophoresis unit for Comet. First of all, in this slide, we're simply looking at the buffer during electrophoresis. We've taken two different buffer concentrations at 150 millimolar sodium hydroxide versus 300 millimolar sodium hydroxide, and the buffer is either at 4 degrees at room temperature. In the panel on the left, what you can see is that as the electrode distance as the distance between the electrodes increases, so does the amperage when you're running at constant volts. The image also shows that if the temperature of the buffer increases or the sodium hydroxide concentration increases, you also have an increase in amperage. The panel on the right is a demonstration of what occurs with the buffer height. As the buffer height starts to increase, you can also see an increase in amperage. So these problems are associated because you're using an alkaline solution for electrophoresis. These problems don't exist when you're using a typical TBE buffer. So when performing alkaline Comet, one problem that frequently came up to our customers is the stated protocols in the literature were using 300 millimolar sodium hydroxide for alkaline Comet. This turned out to be a significant problem with customers who were using power packs that went up to 400 milliamps, which is pretty typical for most laboratories. In order to keep that amperage down, that electrophoresis buffer had to be maintained at 4 degrees in order to keep the amperage down below 400 milliamps. Another issue when trying to optimize your own electrophoresis system is you need to consider the buffer height above the slide. In this example, we're using the Alkaline Comet control cells, and we're looking at a buffer height which is low, which is 0.4 centimeters, versus a slightly higher buffer height of 1 centimeter. So 30 minutes electrophoresis was performed, and then we looked at the alkaline control cells for percent DNA in the tail. So at the lower 0.4 centimeters, you could see optimal separation between the healthy, the C1s, the C2s, and the C3s alkaline control cells. They were all seeing a migration of the damaged DNA out of the tail. However, if that buffer is raised just slightly, the pulling of the damaged DNA out of the tail has significantly decreased.